Hello everyone, my name is Paola Dalgo. I'm doctoral student at the University of Valencia. I want to present you the poster about modulation of TMPO3 gene expression in limb-girdle muscular dystrophy D2. This dystrophy is an autosomal dominant myopathic characterized by severe weakness. The molecular basis was identified as a single adenosine nucleotide deletion at the tag termination codon of the TMPO3 gene, generating a mutant protein with an additional 15 amino acids in the C-terminal domain. TOP3 has been implicated in nuclear import of the proteins, particularly serine, arginine-rich splicing factors. So, what do we know about this dystrophy? Well, LGMDD2 is a relative new muscular dystrophy for which few details of the mechanism are known. Mutant TMPO3 proteins are likely to act as the dominant negatives and interfere with normal function, such as import of the SR family of alternative splicing factors. We hypothesize that reduced TMPO3 mute expression will likely be beneficial in cellular and animal models of disease, in which abnormal upregulate of autophagy is also known, which may serve as a phenotypic measures of enhancement, specifically in degeneration, muscular atrophy, and locomotor capacity. As background to the above, the object of the project is proof of concept using antisense oligonucleotides to modulate the expression of the TMPO3 gene and reduce the synthesis and expression the mutant proteins. For them, we are going to test three strategies using oligonucleotides. The first one is splicing modifiers, which correspond to an intervention in cells processing for which exon directed and exon 23 of the TMP3 gene have been designed to prevent the translation of the mutant protein. The second one, another mechanism via endonucleases RNase H1. At the nuclear levels where ASOs bind to a specific mRNA, this recruits the nuclear enzyme as RNase H1, and the ASOs targets mRNA complex is degraded. And the third one strategy is the use of RNase interference for modification at the cytoplasmic levels, for which we will test the MIR128 that has already been reported to be associated with the modification of TMPO3 by binding to the mRNA slice on the gene, preventing the synthesis of the mutant protein. Our experiments will be carried out initially in HeLa cells, and then the test will be replicated in myoblasts from patients with LGMDD2. The three ASOs strategies will be transfected, and the modulation of TMP3 gene expression will be evaluated by quantification and the protein level by Western blot and the RNA transcription level by RT-qPCR. Finally, as you can see, in the future one, we've designed appropriate oligonucleotide for all three therapeutic strategies mentioned before. We have optimized molecular biology procedures in a reproduced laboratory, and now we are ready to generate data of oligonucleotides activity in cells. Thank you so much.